Hello everyone, Mrs. Hanson once again, picking up with our lessons in chapter 22. It's a Tuesday morning, rather gray outside, so I'm hoping for a little bit of sunshine to pick up our spirits around here. It's been gray for quite a while. So if this is greeting you on a sunny day, let's get celebrating. Chapter 22, Nucleic Acids in Section 2. Well, we know our nucleic acids, there are two varieties, DNA and RNA. Do you see this on the bottom of page 6 or so in your note packs where we've left off together? We're writing out nucleic acids, which are DNA and RNA, really are just joined polymers of our nucleotides that we were drawing in our first lesson together, held together by something called a phosphodiester linkage. I'd really like to take a look at that term, a phosphodiester. See what that term diester? Well, notice here, and I'm just going to put a box around it, a phosphodiester linkage. A phosphate, we had said, determines if our nucleotide is going to be a mono, a di, or a tri, just depending upon how many of those phosphate groups are there. So, for instance, in this particular molecule, we're looking at a nucleotide. And let me just kind of highlight in green. This is one nucleotide. Here would be a second nucleotide. And now they're joined together and we start to create these polymers, which are groups of monomers bonded together. By this particular linkage, one group is bonded to the next. So I know that that looks kind of busy real fast. So let me just make sure we can erase that and start fresh as well. When I'm looking at this first phosphate, this I had boxed just a moment ago, it's linking one nucleotide to the next nucleotide. And the phosphodiester linkage says, notice that I have the 5' carbon in the first nucleotide, and it's connecting to the 3' carbon in the second nucleotide, right? So this is the second nucleotide. And just to remind ourselves, what identifies one nucleotide from the other is the identity of the base. The base in the top nucleotide is cytosine. The base in this second nucleotide is adenosine. So notice that I had those kind of coded for us, cytosine and adenosine. So we see two separate nucleotides being connected by a phosphate group. The type of linkage is known as a phosphodiester linkage. And that means that I have a C-O-P and again leading up P-O-C. We are going to just really emphasize over and over that the 5' carbon of the first nucleotide is going to get to connect to the 3' carbon in the second nucleotide, and the connecting link is a phosphate group. The phosphate group points its carbonyl, the P double bond O, it points it to the left, and it connects the oxygens that are in single bonds, one nucleotide to the next. All right, so we're really examining the picture, and I know that that is highlighted and drawn for you already in your notes, but it's really important to kind of emphasize what we see. The identity of the base would be cytosine. That gives us the nucleotide. And notice that, just as a little quick observation, that I noticed that the sugar has no hydroxyl group at carbon number 2. And remember that it's the anomeric carbon, the carbon that's directly one clockwise position right from the oxygen that's in the sugar cyclic structure. That's where the base is attached, the anomeric carbon. So when I see that this particular sugar lacks the hydroxyl group, I know that this would be a DNA structure, deoxyribonucleic acid. The DNA structure is given to me right away because it's lacking the sugar at carbon number 2. So cytosine and adenosine are the two bases. These are two nucleotides that are joined together. That's why we're calling it a dinucleotide. And we see that a phosphate linkage, it's called a diester because there's two oxygens, a diester linkage, joining one nucleotide to the next. All right, let's keep digesting that. A polynucleotide contains a backbone consisting of alternating sugar and phosphate groups. And that identity comes from the order of the bases, and it distinguishes one polynucleotide from another. So as we begin reading polynucleotides, the identity comes from just distinguishing what base. And remember, I've said it's so important to be able to recognize the structure of the bases. And I said that in my first lesson, that you need to be able to tell me, what does the structure of all of these bases look like? In other words, adenosine, thymine, even uracil, cytosine, guanine, those are the 5 bases. You need to be able to recognize those structures by name. So a polynucleotide has one free phosphate group at the 5' end, and one free hydroxyl group at the 3' end. Can I go back a slide and make sure you see what we're saying? The 5' end has a phosphate group that's free. The 3' end has a hydroxyl group that's free. Now in the joining, 3 connects to 5 when the two nucleotides are joined together. So meaning at the 5' end, there's a phosphate, and down here at the 3' end, there's a hydroxyl group left free. And that's what this is highlighting for us, if you wanted to note that in your picture in the notes there. And then as we look at DNA molecules, the sequence of the bases carries the genetic information of the organism. Remember, as I said, it's the sequence of the bases that as we read to create proteins, the order of these letters matter. Just like creating words, the order of the letters matter. If you have a misspelled word, it would be similar to a genetic mutation where the letters are out of order. So here we have an example, and I know this is also drawn for you, but it's really important to kind of digest what we see. We have a series of nucleotides bonded together. And I see that they're bonded together by a phosphodiester linkage. So I'm noticing that one nucleotide, this is a nucleotide, is bonded to the next nucleotide, bonded to the next nucleotide, all in this series by a phosphodiester linkage. Very important word. The phosphodiester linkage is the phosphate that is attaching one group to the next group. What I've highlighted, or I should say what I boxed in red boxes, those are nucleotides. Boy, did we draw those from lesson 1. Now we're just learning to connect one nucleotide to the next, to the next, to the next, to build polymers. And remember, DNA molecules are millions of nucleotides long. RNA molecules are thousands of nucleotides long. So what connects one to the next is a phosphodiester linkage, a phosphate group. I also need us to highlight that it's the 5' carbon, the carbon that's outside of the ring in one sugar, that is going to connect to the 3' carbon of the next sugar ring. 5' connects to that phosphate, which then connects to the 3' sugar. Over and over again, 5' connects to the phosphate, connects to the 3' sugar. That's known as a phosphodiester linkage. You know what else is kind of cool is how we're just abbreviating the bases by name. So when I see cytosine, I can see a shorthand way of just denoting what those bases are. But when we have to, draw these, you have to be able to draw those bases. And again, when our test arrives, we know that it's a multiple choice test, but you do need to recognize what is the structure called cytosine, what is the structure called adenine. And so therefore, if you see the bases, I want you to recognize them by name. for example, when you see this base we had highlighted on a previous slide, could you tell me that that was cytosine? That's the skill level I need you to get at. And here, can you recognize this structure as the base called adenosine? That's the skill level we need to get at. And so when I think about all of this interesting information, I can tell that we have 3 parts of a nucleotide, just to review from lesson 1, right? We have the sugar, and the sugar is either going to be ribose or deoxyribose. Ribose is a 5-carbon sugar. To distinguish between ribose and deoxyribose, I look at carbon number 2. Does the second carbon, remember the anomeric carbon is carbon 1. Carbon 1 is attached to the base, but it's carbon 2 that would have the absence or presence of a hydroxyl group to indicate if we're in DNA or RNA. I notice that this sugar is missing the OH group. Oop, I hear my dryer ringing. Missing the OH group at carbon number 2. So I now know that this is a DNA strand, deoxyribonucleic acid. Now notice also at the bottom nucleotide, we have a free group of OH. The 3' at the bottom, the terminal end of this polynucleotide, has a free OH group. Up here at the top, the free end has a 5' phosphate group. Remember the carbon number 5, that's out of the ring, isn't it? This is carbon 5 in these structures, and carbon 5 is what attaches to the phosphate group. So we have a 5' phosphate that's free, and we have notice that the interior of this chain has the phosphates, then you see the sugars, and then you have the bases on the interior of this chain. So a phosphodiester linkage hooks together a nucleotide into a next, into a next, into a next for thousands, if not millions long. And notice what this nucleotide would be named. Oh, is that the same CATG? Yep, it is. Okay, so how would we name this? It asks us what's the name of this polynucleotide. How we do this is start at the top at the 5' end, and you read down towards the 3' end, and you just simply list the bases. In order, you're going to list the bases. So we would simply name this polynucleotide as CATG. Now something else interesting, as soon as I see the letter T, the base thymine right here, I now know that this is a strand of DNA because thymine is unique to DNA. T is replaced by uracil in an RNA molecule. So I can also distinguish a DNA from an RNA molecule not only by recognizing that it's lacking the hydroxyl group at carbon 2, that's the DNA sugar, but also by seeing the presence of thymine, the base that is unique to a DNA molecule, is a second giveaway that I would see knowing that this is a strand of DNA. So you have a sugar phosphate backbone, the phosphates and then the sugar, and then on the interior you see a sequence of bases. You read them always from the 5' to the 3' end when we're naming the base. Are you ready to practice? It says let's practice. Let's draw a structure of a nucleotide forming by joining the 3' end hydroxyl group of AMP to 5' phosphate in GMP. We're going to label the 5' and 3' ends and then simply name the dinucleotide. Okay, so let's, one step slow. First of all, we better figure out, let's interpret what does AMP stand for and what does GMP stand for. And that goes back to last lesson, right? These are nucleotides. A will stand for adenosine, and then you have 5' monophosphate. Adenosine 5' monophosphate. That's the name of this nucleotide. How about GMP? Well, that is called guanosine 5' monophosphate. So we really have just started by recognizing if we got to hook together two nucleotides, let's understand what the two nucleotides look like. So I am going to take the AMP, and let's just start by drawing the 5-carbon sugar. 1, 2, 3, 4, 5. That's a 5-sided ring, and then the 5th carbon, of course, comes out here, CH2. Now, let's decide the sugar, okay? The sugar, well, I don't see it written as DAMP, and I don't see DGMP. I don't see this little D in the name of the nucleotide, the D standing for deoxy. Since it's absent, I know that the sugar must be the ribose sugar. So I know that it's going to have, it's going to have an OH group at carbon number 2. For now, I'm just going to abbreviate and just kind of get my bearings straight here. I'm going to abbreviate the letter A to represent the adenosine base. And I know it's going to be attached to the anomeric carbon at carbon 1. But for now, I'm just going to put a letter A and come back and fill it in. I just want to get my sketch kind of going. Carbon 2 I know has an OH group because it is a ribose and not a deoxyribose. Here's carbon 3, carbon 4, carbon 5 in my sugar. All right, the ribose sugar has 5 carbons, where the 5th carbon is out of the ring. And notice that the 5' carbon is going to have a free phosphate group. And so at this point, I know that the phosphate group is going to be unattached. So I can just put that there, knowing that that's going to be the terminal end of this, would be a free phosphate group. At carbon number 3, we have a diester phosphate linkage, which is going to then hook to the carbon that's in the 2nd nucleotide. And notice that the double bond gets placed to the left and the O negative still down below. I've just drawn the phosphodiester linkage, and I'm leading into the next sugar. Oops, I should make that come in. Does that look like a 5-sided sugar? I could probably do better. I can make that come up to the oxygen, back down. That's a little better, isn't it? And so for now, at the anomeric carbon, I'm going to just put the letter G for the base guanine. Alrighty. And then I know down here at carbon number 2, there's going to be at carbon 3, a free OH group, and at carbon 2, I need another OH group. My phosphodiester linkage is right here. That's the phosphate group that's joining together these two nucleotides. This nucleotide is GMP. It has a free 3' end. Right here is the 3' end. This nucleotide was AMP. It has a free 5' end. That's the phosphate group. So here's the 5', here's the 3' end. And to name this dinucleotide, we just simply put the letters from 5' down to 3'. So this would be called AG. Now, really what I need to do is draw these bases. And let's take that step because we're not yet done. We're just kind of going in pieces. So let me kind of review what we did. This is the same question, just kind of going through it by step again. So we're asked to draw the dinucleotide. That means we're going to have just one phosphate linkage hooking together two separate monomers called nucleotides. The first monomer is an AMP, and I said we need to understand what AMP stands for. And I labeled you as adenosine 5' monophosphate. That's AMP. Here's what we need to recognize as adenine. We know that it is a 2-cyclic ringed base, and that the major functional group up here in the second ring has an amine. The second one that we were labeling is GMP, and I said we need to understand what that stands for, and that's guanosine 5' monophosphate. That's GMP. And we have to be able to recognize that guanine is also a 2-ringed base. Making its structure unique is a carbonyl ketone, C double bond O, and another structural amine group out here. We recognize that the sugar would be ribose because we did not hear little d in the name. So therefore we know that the sugar is ribose. That's how I knew to draw on the OH at carbon number 2. Carbon 1, the anomeric carbon, serves as the site of attachment to the base. Carbon number 2 will have a hydroxyl group because it is an RNA molecule. Carbon number 3, carbon number 4, carbon number 5 is up out of the sugar. Putting those pieces together, here's what we found. Let me make this bigger for us. We said when we draw the sugar, remember how I took a shortcut, and for this right here, I just put the letter A. And when I did that, I was going to come back and actually draw in the structural unit there for adenosine. But when I start assembling pieces, I usually will take a shortcut and just get the bases written by name by just writing their first letter and then go back and fill in. And then here, same story. A moment ago when we drew together the rough draft, I drew in the letter G to represent guanine, and then I do need to go back and actually fill in that structure. We wanted to highlight that the 3' end and the 5' end come together to create the phosphodiester linkage. And that's this highlighted in blue now is the phosphodiester linkage, that the phosphate joins the 3' end of that sugar above it. And when we label this, we just said you're going to name it as AG. Just name the bases as you go down from the 5' end at the top, reading down to the 3' end. Now, make sure in your notes you've taken the time just for repetition, because that's how we learn. Repetition's our friend while we're learning. And make sure that you've drawn in adenine and guanine bases. All right, that's a lot of drawing. How did you do? This is GMP, guanosine 5' monophosphate down here. And of course, up here was AMP. That was adenosine 5' monophosphate, hooked together by one phosphodiester linkage. Whew, all right, let's try another. Draw a polynucleotide called TAG. What does that look like? TAG. Well, T, thymine, A, adenosine, G is guanine, TAG. Draw the polymer. Well, let's decide first of all, TAG. So thymine, adenosine, and guanine. Those are the bases, and this would start up here, knowing that this would read from 5' down to 3', because that's how we're naming TAG. What about the sugar? Well, since we were not given the names of the nucleotides specifically, we have to be a little bit of a detective and decide, is this going to be a polynucleotide of a DNA or a polynucleotide of an RNA? And the reason that we can know that is by the presence of the base called thymine. That thymine is unique to a DNA molecule. DNA has thymine, RNA replaces thymine with uracil. So here I know for the sugar to be deoxyribose sugar. Now, what I'm about to do is just a rough draft, all right? This is how I get started. The first thing I do is to just simply begin drawing my sugar as a 5-membered ring, and then I know that the 5th carbon comes up, that's CH2. And I know that the anomeric carbon holds the base. And just for now, I'm going to abbreviate the base with its letter T. And down here at the 3rd carbon, 1, 2, 3, 4, 5, all right? At carbon 3, I know that that's going to be the phosphate linkage. So how about just for now, I'm going to put P for phosphate. And I know that this guy will have a P for phosphate, because the carbon that's number 5 joins together the 2 nucleotides. So I just drew a rough draft, if you will, of the first nucleotide that would be up here at the 5' end. And that would be the nucleotide that contains the base thymine. Now, the phosphodiester linkage is going to join thymine to adenosine. And so I'm going to draw its 5-membered ring sugar, knowing that the base at the anomeric carbon this time is going to be adenosine. I'm just going to abbreviate it A for now, and then I know I can go back and look up its structure, and I can come back to the details. And I just also remember that at carbon 2, I purposely left off the OH group because we have deoxyribose sugar. Alrighty. And so now I can take this carbon, which is carbon 3, and I'm going to attach it. Let's see, TAG. We have to do a G yet, don't we? So this is going to be a phosphate linkage, attaching carbon 3 to carbon 5 of the next nucleotide. And I'll make its sugar a 5-membered ring, knowing that it's going to have a G, guanine, for a base. And just for now, let's write G, and I know I can come back and fill in the rich details of the structure. Carbon number 3 this time is going to have a free OH group. So the 3' end has a free hydroxyl group. The 5' end up here had the free phosphate group. And I don't put OH in at number 2 because we're in a DNA molecule, so it's left without the OH. All right. So you see how this is just kind of a rough draft. It's a sketch to get going. I have 2 phosphodiester linkages. Alrighty. So when I have 3, nucleotides, I'm going to need 2 linkages to connect those 3 molecules. T needs to link to A needs to link to G. We have this rough draft. We're ready to fill in the details. For instance, this phosphodiester linkage, the P leads out to both of those carbons. So here is the carbon that would be in the sugar. This is the carbon that's carbon 5. We end up with a double bond leading to the left and a charged oxygen leading down. So just kind of thinking about what this box with a P inside of it means, that it really is standing for the structure that has the oxygens connected to those carbons. You'll have a double bond, and then this guy will be charged. That's exactly what I mean when I have these in the boxes there. Alrighty. And of course, if it's a free end, then I have just like this then. You have both of those oxygens connected then. So the 5' end has 2 charged oxygens. Alrighty. Now let's just take that up a step and kind of fill in some of those other details. We said we have to begin really by identifying the polynucleotide, TAG. So T we know stands for deoxythymidine 5' monophosphate. It comes from the base thymine. So here is thymine. A moment ago, I just had a T in a box to represent that base. I have to be able to recognize that structure as thymine. The next one we had in that sequence was adenine. So a moment ago, I just had A in a box, but I know that this is what I have to draw in for details. The same with letter G. This is guanine. Remember the skill level I'm asking you. The skill level is to recognize the base. So if you see the picture of thymine, you know to name it thymine. You have to be able to look at the picture and identify it by name. Alrighty? Because here's the skill. Like if you are asked to draw TAG, I know you can look up the structure and draw it. But on a multiple choice test, even on your quizzes, you have to be able to recognize the structure without having that, the little piece of notes in front of you there. Look at the structure and go, okay, that's thymine. Look at the structure and go, okay, that's adenine, and same for guanine. There are 5 of those to be able to recognize by name. And I mentioned we knew that it had to be in a DNA molecule because thymine is unique to DNA. That's how I knew to draw the sugar. And when we put the whole thing together, this is what the final structure looked like. So pause and draw in with ample detail the following. Instead of having just a box of T, take the time to actually draw in the base, thymine. Instead of having just a box with the letter A, take the time to draw an adenine, and the same for guanine. Thymine is a 1-ringed base, but adenine and guanine have 2 rings in their structure. The anomeric carbon is the site that attaches to the base. In each one of these nucleotides, there are 5 carbons. 1, 2, 3, 4, 5. Notice the 5' end has a free phosphate group. And the 3' end has a free hydroxyl group. I'll highlight those. The free phosphate at 5' and the 3 hydroxyl at the 3' end. When you name polynucleotides, I didn't need the ruler. When you name them from 5' to 3' N, so the name is just TAG, naming them in this order, 5' to 3' end. The phosphodiester linkages connect the 5' carbon to the phosphate up to the 3' carbon in each of those. So there we said there were 2 phosphodiester linkages connecting the 5' carbon of one nucleotide to the 3' carbon of the next nucleotide. That's a phosphodiester linkage. Whew. All right. So that was our second section in our chapter. What we're looking now is kind of a bigger picture. So let's suppose we start putting some of these polynucleotides, TAG, UC, we get all of these letters in a row. What does it start to look like in a macro scale? Well, the current DNA molecule was originally proposed back in the early 50s by Watson and Crick. These 2 gentlemen came up with the alpha helix proposal, the polynucleostrands of a DNA molecule. And they twist, just like ribbons twist a rotating ladder, if you will. The two strands do run in opposite directions. So one will run from the 5' end to the 3' end. The other will go in the opposite direction, 3' to 5'. So if you think about the rungs of a ladder and just kind of thinking about that through, this would be the 5', 3'. Its complementary strand runs the opposite direction. And in a moment, we'll see a next slide where those two bases are going to have hydrogen bonds holding them together. And then you just twist this to come up with the idea of a alpha helix. Now, on the outside, this is your sugars and your phosphate backbone. And the bases line up interior. They lie on the inside of this two-runged ladder theory. So let's take a peek really at an alpha helix, kind of the helix that makes up the DNA molecule. Okay, so here's some things that we said. From 5' end to 3' end, this would be the first strand of the DNA molecule. Notice that the phosphates and the sugar lie on the outside, and the bases lay in the interior. In the next strand, we go from 5' up to 3' in this direction. So they're the complementary order. So 5' down to 3' this way, but this is going in the opposite direction. Here's another thing to notice. The bases that are in the middle have a very specific arrangement. For example, adenine is color-coded in green here. See, this adenine is a base. Adenine, we've learned, is a two-ringed base structure. So here's another adenine, color-coded green. 2 rings in its base structure. Do you notice that adenine always bonds to thymine? Thymine is a 1-ringed base structure. Here's another thymine, color-coded here in pink. It's a 1-ringed base structure. I noticed that A bonds to T always, and that this base has 2 rings and this base has 1 ring always. Do you see how many hydrogen bonds are holding together an A to a T? 1, 2. 2 hydrogen bonds holding those together. So one strand of the DNA molecule being held together by 2 hydrogen bonds to the other strand when an A lines up to a T. I was writing bonds and said the wrong letter. What else do we see? Kind of color-coded in, I don't know if I have the right color. Color-coded in kind of a orangish maybe. I can make it orange. We have this particular molecule. Color-coded in orange is cytosine. Trying my best, that's orangish. Cytosine. And what I noticed that it's kind of color-coded next to is blue, or purple, I guess, but I can make it purple. Here's purple, and that is guanine. And so I'm noticing that the cytosine and guanine are always hooked together. So a G attaches to a C, always. G goes to C. The guanine has 2 rings. The cytosine has 1 ring. That 2-ring bases will always bond to a 1-ringed base. How many hydrogen bonds do you see lining up with this pair? Well, I see 3 each time. So when G aligns with C, they can form 3 hydrogen bonds to hold those bases together. When the bases align, a pyrimidine derivative can hydrogen bond to a purine derivative. A 1-ring base can align itself with a 2-ring base and hold together by hydrogen bonds. There is always a complementary base pairing that occurs. When A binds to T, C goes to G, always, because we have a 2-ringed base combining with a 1-ring base in each one of those complementary base pairs. Adenine pairs with thymine, and we notice that it did so with 2 hydrogen bonds. I always remembered it by just putting a little equal sign, A equal T. Adenosine, 2 hydrogen bonded to thymine. And when C joins to G, C to G, it does so with 3 hydrogen bonds, forming a base pair. C always goes to G, A always goes to 2. And so if we call the original strand the 5' end to the 3' end, and that's how we always read the code, right? When you are reading, and I mean reading the code, just say naming the polynucleotide. That's a nucleotide. You always read from 5' to 3'. And so I would name the original strand as TAGGCTA. I'm just telling you the basis: thymine, adenine, guanine, cytosine, thymine, adenine, on and on and on. Each one of these letters is giving me a nucleotide. So the complementary strand reads it in the opposite direction, right? Here's the complementary strand. 5 complements the 3' end. The 3' end complements the 5' end on the complementary strand, and you just create the complementary code by reading what the base attaches to. T always attaches to A goes to T, G goes to C, and we come up with the complementary base pairing. Notice that when we really examine closely, here is a thymine base. Notice that I just kind of drew squiggles here, just showing that that's going on then to the sugar that's attached to it. So it's just kind of a shorthand to say these are the sugars, but I'm just examining where the bases from one strand ally to another. So this would be like the 5' to 3' end, and you see this backbone going on. This is the 3' to the 5' end, and you would see this backbone, and the bases lie in between. What holds one strand of a DNA molecule to the other are these very strong intermolecular attractions known as hydrogen bonds. When an A and a T bond together, clearly we see 2 hydrogen bonds between those complementary bases. And when a G bonds with a C, the GC base pairing, we have 3 hydrogen bonds that are holding the 2 ribbons together. And the molecule itself is said to be a twisting ladder, an alpha helix. Notice that G pairs with C, T pairs with A, up and down the ladder we go, and we get the complementary base pairing. The sugar phosphate backbone is represented by this ribbon, and the 2 ribbons held together by base pairing. And this is just another closer examination when we see the rungs on this ladder. When G binds to C, we have 3 hydrogen bonds. And when T bonds to A, you get 2 hydrogen bonds. That's what's holding the ribbons of the ladder together. You get an idea of how often my email goes off. Let's practice just kind of getting an idea of what complementary base pairing means. Write the complementary strand for each of the following strands of DNA. So I just want you to think about 5' reading to 3'. The complementary strand has to go the opposite direction. So 3' to 5'. What would the letter codes be? Well, I can do the first one with you. I know that A always pairs to T. So AAA gets coded TTT. C goes to G. G goes to C. T goes to A. C goes to G. C goes to G. We just wrote the complementary strand for the first DNA molecule. How about the next one? T pairs with A. A pairs with T. T pairs with A. A goes to T. C to G. G to C. C to G. And again G. Pretty easy, right? We've got this. A goes to T. T goes to A. A goes to, or T goes to A. C to, I'm mixing it up. I'm going too fast. G goes to C. I write C. Here's T. G. 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 C. G. Whew! We did it. This enormously large DNA molecule that composes the Human Genome Project, the total DNA content of an individual, if we just kind of consider how it's put together, there's nothing to write here. It's just kind of a fascinating thing. This is where we were just talking about, is these individual base pairing of the alpha helix DNA. And as it begins to twist and twist and twist, it wraps around a structure, and that structure called histones, which are protein molecules. And the histones themselves begin to wrap around and create this chromatin fiber. This chromatin fiber begins to just spiral, 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 spiral until it creates individual chromosomes inside the nucleus of the cell. And there would be 23 pairs of these chromosomes in humans that give us our genetic makeup. So our question is, how is this information stored in DNA molecules, and how do we use it to direct the synthesis of proteins? I'm going to just pause and kind of let us digest that and come back in a next lesson.